Now we are moving to the section enrichment analysis using a ranked gene list. Now that we have seen the workflow for a defined gene list, let's see the workflow for a ranked gene list. Why test enrichment in a ranked gene list? If you're able to rank your genes, the ranked gene list approach is always recommended over the defined gene list because we are trying to avoid arbitrary threshold to select our genes. It is difficult to know where to put the threshold to select genes. You could be too stringent, and in this case, you are going to lose information, or at the contrary, you could be too permissive and allow too many false positives. But with the rank list, you are avoiding this issue. For ranked gene list, we are taking bulk RNA-seq as example. The reference paper for this data is below. In this example, we overexpressed a transcription factor in blood cell, and we compared the control and the treated cells using RNA-seq. So OE means overexpression. From the results of the differential expression analysis, we derived a rank list ranking all genes from the genome from top up regulated to down regulated, and calculated enrichment analysis using the tool GACA. Then we visualized the results as a network, as we will explain to you in the other modules, and focused our attention on two particular pathways, MYC and lysosomes. We are going to go through how GAC works in the next slides. How do we create a ranked list? Let's take the example of RNA sequencing. In this example, we have two groups of samples, like treated and control. Let's say treated, control, class 1, class 2. We use the R package HR or DSEQ2 to estimate the differential expression of each gene. Some genes will find significantly differentially expressed and some not, but we have the results for all genes, and we will keep all genes in the rank list. We will have the genes upregulated in group 1 compared to group 2 at the top of the rank list, and the genes downregulated at the bottom of the list, and all genes that do not change their expression, so the non-significant genes will be in the middle. Whether we use HR or DSEQ2, both of them give us the output table, the value of the log fold change, and the p-value. And this is what we are going to use to derive a score that we are going to use for the rank list. So the formula to create the score is indicated above the table. This is sine of the log fold change multiplied by minus log 10 of the p-value. We only take the sign of the log fold change plus or minus because it indicates us if the gene is upregulated or downregulated. And we also calculate a score using the value of the p-value. So the transformation minus log 10 of the p-value will give a high score for genes that have a very small p-value and then are strongly differentially expressed. This way we can rank all genes from top up regulated to top down regulated. We can do it in Excel or in R. So GACA, it is Mutha et al. Who developed GACA in 2003. They were studying diabetes. They came out with the GACA algorithm that showed the downregulation of the oxidative phosphorylation pathway in their model. The particularity of this finding was that the genes of this pathway were not strongly downregulated. They were all downregulated by a small amount, but not significantly. But the addition of all these subtle amounts had a strong overall effect on the pathway, and that could be captured by the GACA method and then further experimentally validated. The statistical test used by GACA is a modified Kolmogorov-Smirnov test, KS test. Now we are going to see how the GACA running sum and enrichment score are calculated. Our rank file is on the left with upregulated genes at the top and downregulated genes at the bottom. So we place this rank file horizontally with the up genes on the left and the down genes on the right. The genes that are not significantly differentially expressed stays here in the middle and will not contribute to the enrichment scores. The black bar indicates genes that are included in the pathway that we are currently testing, and we can see that there is like a higher density of these genes toward the left side of the rank file. It means the pathway enriched in our upregulated genes. The GSEA running sum starts at 0 and at gene 1 of the rank list, and then it goes gene by gene, and each time a gene is in the rank list, is in the pathway, then the sum increases a little bit. And each time the gene in the rank list is not in the pathway, the running sum is decreasing by a very small amount. Therefore, if you have a higher density of genes in the pathway at the top of the rank list, the running sum is going to increase rapidly to reach a maximum and then decrease where there is no more genes from the pathway. The maximum that has been reached in the running sum, the peak point that I'm showing here, is described as the GSEA enrichment score. GSEA has a weight system, which gives more weight for the genes that are at the very end of the rank list and less weight for the genes that are considered non-significant and that are located in the middle of the rank list. So you can have peaks on the left or peak on the right, but never peaks at the middle of the rank list. This is a zoomed image that shows us how the running sum, which starts at 0, increases when a gene from our list is in the tested pathway and decreases slightly when there is no genes from our list that is in the pathway. Then we understand that we need more genes in the pathway at the top of the rank list for a peak to be formed. Pathway can be enriched in genes upregulated. In this case, the enrichment score is going to be positive, left side, or the pathway can be enriched in genes downregulated. And in this case, the enrichment score is going to be negative. Now that we have calculated the enrichment score, we still need to estimate if that score is equal or larger than the one that we could have obtained by chance only. GSEA method is using permutation to build a null distribution and to calculate the empirical p-value. In the case that we are using most of the time, the permutation is done by replacing the genes in the pathway by random genes. But there is another permutation technique that consists of shuffling the samples before creating the rank file and basically creating 1000 random rank files to break gene dependency. It's called label permutation. For each tested pathway, we have the enrichment score for 1000 permutation. That's the null distribution that you are seeing on the left that should be centered around 0. And then we have the observed or the real enrichment score. So now we can calculate the distance of our observed score from the mean of the null distribution. If our score is far away from the center of the null distribution, it means that this score does not occur frequently, therefore it's unlikely to happen by a random chance only. GSEA also calculates the FDR value from the normalized enrichment scores. So we have now finished this part on GSEA, and we will see in the next part examples of ranked and defined gene lists. So our mixed gene list, ranked or not ranked. We are going to see it through an example so you can choose it for your own data. So here are some examples of our mixed data for which you can generate a ranked gene list. The first one is the bulk RNA-seq. We calculate differential gene expression between our two conditions, let's say treated versus control. We rank all genes using the differential expression score from top up regulated to top down regulated. The second example is for single cell data. We obtain clusters defining two groups of cells, and then we calculate differential expression of these two groups, and we can generate a rank file. Finally, label-free proteomics can lead to a sufficient number of proteins to create a rank list. When we compare two groups, treated and control, and we use the same ranking strategy as for the bulk RNA-seq. Now, some example where we more frequently use a defined gene list. Even if a ranked gene list is sometimes recommended, there are some cases where we cannot create this ranked list. In the first example, the starting point is DNA. We are looking for somatic mutations or copy number variants in a cohort of patients, and our gene list will be the genes that are frequently mutated in our patients. Here's an example of another experimental design, and it's a time course, for example, developmental biology. We have collected our samples at different time points, and we are going to use some clustering algorithms to group the genes that have the same profile. Therefore, each cluster or group is a gene list. We will perform enrichment analysis individually on each gene list. The third example is enrichment analysis of attack or ChIP-seq data. Peaks are detected, and if this is a two-class experimental design, like a control and treated, we can select the peaks that are unique or more intense in the treated condition. We have then defined a list of peaks, and we will retrieve the genes near the peaks to create a gene list. For all these cases where we have a defined gene list, we will use the Fisher's exact test to estimate pathway enrichment analysis. As I said previously, we are presenting you a few tools during this workshop, but many tools exist for enrichment analysis. These tools can be web-based, like GProfiler, included in a Cytoscape app like Clugo, in a standalone application like GACA, or in an R package. What we have learned is that a typical output for enrichment analysis would be the list of the tested pathways, a value related to the size of the overlap. It could be the overlap size of the enrichment score, the number of genes in the pathway, or the name of the genes in the overlap, a p-value, and very importantly, a corrected p-value. Well, not to mention that many pathways in this output table will contain genes in common because they are functionally related, but it's difficult to see in a table format. That's why network visualization of this result is going to be important. Now that we have covered the theory, here is a list of questions that could help you when you need to choose a tool for your analysis. So, does the tool cover your model organism? Is there a good choice of gene sets? Are the pathway database up to date? Is the tool up to date? Which statistics do they use? Is it for gene list, rank list, or do you have the option to choose one or the other? Is this description of the statistics clear enough? So for this point, you usually should go to the original paper and see if you understand the statistics. Do you like the output style? Can you connect it with network visualization like Cytoscape, or do they offer like a small network to visualize the results? So here we present like different tools like GProfiler, Panther, Bingo, and Clugo. And GProfiler is the one that we are choosing to show you during this workshop. So does GProfiler have updated database? Yes. More than one pathway database to choose from? Yes. We can use this database individually or together. So there are different options available. Does it cover multiple organisms? Yes. We can upload a custom database or a custom GMT file, custom pathway database. The statistical test is a Fisher's exact test. It corrects for multiple hypothesis testing. There are actually multiple methods for this. possibility to upload the custom background. So for example, if we have a nano string and a restricted background, we can do that. So it's a web-based, and it also can be connected with Cytoscape. So we have finished the gene list enrichment, and as a last note, we are looking for over enrichment of pathway, but in rare case, people are looking for under enrichment of pathway, and it's possible to do it as well. And keep in mind when you are looking for enrichment tools that the Fisher's exact test is often called a hypergeometric test, and that other similar enrichment tests for gene list are approximation of the Fisher's exact test, sometimes using Monte Carlo simulation, binomial test, or the chi-square test. So this is two tools for the rank list. So GACA that use a modified KS test, and Penter that use a Wilkinson rank sum test. GACA correct for multiple hypothesis testing. We can upload different database, so we can it's a custom, but we can use the database of our choice. And yes, we can connect it with the Cytoscape software so we can visualize the results as a network. So I mentioned the Wilcoxon rank sum test, and it's another test that we could have used when we have a ranked list. It's non-parametric and it only consider the ranks of the genes. It's the same as the idea as GACA, where if the pathway is enriched in genes upregulated, we should see a shift in the distribution compared to the null distribution. So we can see in red, we have the distribution of the tested pathway, and we see that it's shifted to the right because the genes in this pathway are upregulated, and it's compared to the background population, which is in blue. So this ends this part 3 of the lecture.